Welcome back to Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. What I wanted to do in this video was do a very basic overview of how someone would be tested to determine whether or not they actually are infected with COVID-19. Okay? And it turns out that it's going to involve a test that uses this principle of real-time quantitative real-time PCR. Now, for those of you in the molecular bio community, this might be much more familiar, but I want to actually break this down in the basic terms so that anyone can understand it. Okay? In one of the previous videos, I mentioned that the COVID-19 virus is an RNA virus. Okay? So obviously you know that we as humans and most other organisms have DNA. That's our genetic information. Some organisms, like viruses, can actually have their genetic information contained in RNA, which is a closely related cousin, so to speak, of DNA. So the COVID-19 virus is an RNA virus. So if we want to detect whether or not somebody's infected with the virus, one good way to do that would be to test for that viral RNA. Okay? And so the way we're going to test for that viral RNA is through a process called quantitative real-time PCR. Okay? Now here's a general workflow of what would happen if somebody came in with a possible infection and you wanted to actually test for it. Well, you'd of course have to collect a tissue sample, okay? And that tissue sample presumably would contain the viral RNA, it would contain the virus and the viral RNA. And then you do some processing and you isolate that RNA. So you do what's called an RNA extraction. RNA is a type of nucleic acid, so you extract all the RNAs. Now in that sample, you're going to have the human's RNAs, the patient's that is, and you're going to have other RNAs like the RNAs of maybe some bacteria that are there. And then somewhere in there, you're going to have some of that viral RNA, presumably, if the person is in fact infected. Now out of all of those RNAs, how do you get to the needle in the haystack? How do you get those little viral RNAs out of all the multitude of RNAs that you would have in that sample? Well, you specifically amplify the viral RNA, but the way that it's generally done is you convert the viral RNA, COVID-19, into DNA, okay? And this is cDNA or complementary DNA. So it's DNA derived specifically from the RNA that came from that virus. And then once you have this cDNA, which is done through a process called reverse transcription, then you amplify that DNA and you'll get something that looks like this. Okay? Now here's the problem. If we're looking at DNA or RNA, and I say looking very loosely, they're microscopic. You can't see them. If you try to put it under even a really powerful microscope, you won't see it. Okay? So how do we see RNA or DNA? Well, to do that, we need to attach the DNA or RNA to something that we can see, like something like light, something that's fluorescent. What I really want to focus on, though, is I want to focus on this amplification of this viral cDNA by real-time PCR. And the reason I want to focus on this is actually what's going to allow the machine that measures this to ultimately determine how much of the DNA there was in the sample. And we need to think about this going backwards. Okay? If I were to amplify this cDNA, this viral cDNA, and I get a lot of it through the amplification, that means there was a lot of this viral DNA to start with. And if there's a lot of this viral DNA to start with, that means before that there was a lot of viral RNA. And if there's a lot of that viral RNA, that probably means that this person was infected with that virus. Okay? So I'm going to go into a little bit of detail in the next slide, and then we'll come back and look at the big picture and kind of look at this and see what this means in the grand scheme of things. So once we have that viral DNA that we've made through reverse transcription on the viral RNA, we're going to perform an amplification. This is the real-time PCR amplification. So when we amplify DNA in PCR, we have to use primers. Okay? Now for molecular biology people, this is understandable. Basically what the primers do is it allows us to amplify specific regions of the DNA. So for instance, if we wanted to amplify only the viral part, then we would pick primers that are complementary to only the viral DNA, but not our own DNA. Okay? So we amplify the DNA with these primers. But before we do that, we mix the DNA, that is the viral DNA, with this little probe right here. Now the F stands for a fluorophore and the Q is a quencher. Okay? Fluorophore is something that when it gets by itself, it fluoresces. Basically, it's going to release some light, and we'll be able to detect that light. But as long as this fluorophore is bound to the quencher, it will not do that. Quencher, like quenching your thirst, it stops your thirst. In other words, this quencher stops the fluorescence from happening. And as long as this probe is intact in this form, this fluorophore will not fluoresce and it will do nothing basically. But as soon as we start the amplification, we're going to start amplifying the DNA. We're copying it on both sides. And when the amplification process gets to this probe, it basically splits off the fluorophore. And now the fluorophore is no longer connected with the quencher. And so basically what this means is that every time we see fluorescence, that's indicative of an amplified piece of DNA. And so overall, the more fluorescence we get, the more viral DNA we had, and like we said, the more viral DNA we had, the more viral RNA, and therefore if we have a lot of viral RNA, it means the person was most likely infected. Okay? Now this graph right here is really focusing on that last step. This is the amplification of the DNA, the PCR, with that fluorescent probe. And so if we look at this graph, on the vertical axis we have the fluorescence intensity. So as you go higher up on the vertical axis, that means there's more fluorescence, and that means more DNA. On the horizontal axis, this is just the cycle number in the amplification. Generally speaking, you can use any amount of cycles you want. But according to the CDC, and I'll show you this in a few minutes, the CDC's protocol says to do exactly 40 cycles, so that's why I have it from 1 to 40. And so as we're going from 1 cycle to 40 along this horizontal axis, if a person had that viral RNA originally, then we would expect this curve to increase, and then eventually it'll come to a plateau like this. Now what I'm going to do is I'm going to try to explain how this works by looking at this red curve, and understand the blue and green ones are going to work in a very similar manner. But before we do that, let's talk about something called the cycle threshold, C sub t. The cycle threshold is this dotted line right here, and this is kind of like a minimum detection of the instrument that's measuring the fluorescence. Okay? We obviously don't just look at it and determine the fluorescence. We use an instrument to quantify it absolutely. So it turns out that whenever there's a very, very low amount of fluorescence, the machine's not really good at determining whether or not that's just error or whether there's actually fluorescence. And so we have to get to a high enough point in the fluorescence where we can really be sure that there is an increase in fluorescence. Below that point, so below the cycle threshold, we can't be sure that any of this below that is actual increases in fluorescence. It could just be error in the instrument. Okay, so just random error. But above the cycle threshold, that's the point at which we can start to assume that the increases in fluorescence are actually increases in fluorescence. Okay? And so as we're amplifying that DNA and we start releasing more fluorophores, more fluorescence means more amplified DNA. And so what we see here is that as we go along the cycles for this red curve, let's say this is the viral RNA, we start to see an increase in the curve. It's going up and up and up. What that means is that the fluorescence intensity is increasing. That's the vertical axis. There's more fluorescence. So that means we're seeing more and more DNA, and eventually it gets to a point where it plateaus, where the machine really doesn't detect any more fluorescence. Now what does this mean? It means that because we're seeing all this fluorescence, that means we have viral DNA. If we have viral DNA, we had viral RNA before that, and if we had viral RNA, we likely had the virus, and there was an infection. Okay? Now what you see here are 3 different curves that look almost the same, except that this blue one is shifted after the red one, and the green one is shifted after the blue one. If we think about what that means, it means that this blue one, whatever RNA this represents, it didn't start getting a lot of increases in fluorescence until well after the red one did, and this green one took a long time to get increases in fluorescence. But notice that both of them did increase above the cycle threshold. They just both took longer. What this means when you look at a graph for quantitative real-time PCR, assuming that you have a bunch of curves like this that all increase above the cycle threshold, and they'll generally all look very similar, the one that increases above the cycle threshold last is the one where there was the least RNA. And so what we can say is that in the original sample, whatever this was, we had the most of the red RNA, the least of the green, and then somewhere in the middle for the blue RNA. Okay? So yeah, in this sample, the red sample had the most RNA, while the green sample had the least. And because the green one was the least originally, that's why it took so long to see that increase in fluorescence, which means increase in DNA, at the amplification step. Okay? And so it doesn't mean that there wasn't any RNA, there clearly was. If you had a sample where there was no RNA, you would never see these curves increase above the cycle threshold. So if there was ever a situation where it just didn't go above the cycle threshold, that could conclude that there was no RNA in that sample. Okay? And therefore, if there was no RNA, there wouldn't be any cDNA, and there'd be nothing to amplify. And that's why you wouldn't see anything above the cycle threshold. Now let's do an example here, real-world example, where this red curve represents the original viral RNA. Now understand it's DNA that's being measured here, amplified with the probe, but if you have this curve that increases like this means that there was viral DNA amplified, which means there was viral RNA, and you do have the infection. Okay? Now, in the analysis that's required by the CDC, it's a little bit more complicated than that. So what I'm actually going to do is I'm going to go to this diagnostic panel right here. You can find this on the CDC website, but here's a link to it. I'll try to remember to put that in the description of the video. And so here's actually the PDF. You can actually find this. It's on the CDC's website. I'm actually going to scroll down here, see if I can find it very quickly. So here we have 2019 nCoV markers N1 and N2. The original RNA genes that you're looking at from the virus, there's an N1 gene that we're looking at from COVID-19 and an N2 gene. So there's an N1 RNA and an N2 RNA. They're both converted to that DNA, and then they're amplified with the probes to get all that fluorescence, and you would see something that looks like this. Okay, so the first criteria here is when you have all controls exhibit the expected performance, a specimen is considered negative, meaning it doesn't have the virus, if the viral gene curves do not cross that cycle threshold. Okay, so if we had a situation where we had a control that behaved as expected, let's say this green one was the control. Okay, you have to have a control so you know whether or not the experiment worked. But these two represented the virus and they didn't increase above the cycle threshold, then you could conclude that the person is not infected with the virus. There's a table of this on the next page right here. So for example, if the control, which is the RP here, okay, that's actually human gene RNase-P, okay, if this was actually positive, but both of the viral genes were negative, then you would conclude that the virus is not present. The virus is not detected and the person is not infected. Okay. So scrolling back up here, however, if all the controls exhibit the expected performance, a specimen would be positive for the virus if those two viral genes did cross the cycle threshold, but it has to be within 40 cycles. So in other words, if you had something that looked like this, where let's say the green one's the control, it worked clearly, but then you also have the two viral RNA genes, and those curves both went like this, then this would be indicative that a person is infected with the virus. Okay? And so back to this table, if the control worked, as you can see right here, and then both of these viral genes went above the cycle threshold, then the person is positive for the coronavirus, okay, COVID-19. It's important to notice that when you're looking at this positive result, however, the control actually doesn't have to work. Even if the control comes back negative, as long as both of these viral genes came back positive and across the cycle threshold within 40 cycles, the person would still be positive. Now here in the second row, regardless of whether or not the control works, it can be positive or negative. If the person tests positive for one or the other, so not both of them, but maybe they test positive for the first COVID-19 gene and not the second, or vice versa, if it's only one of them, it doesn't matter if the control works or not. The test is actually inconclusive. So to get an absolute positive result for the COVID-19 test, you actually have to test positive for both of these viral genes, N1 and N2. And then at the bottom right here, if the control is negative and both of the viral genes are negative, that doesn't necessarily mean that the person is free from infection. It's actually an invalid test. Okay? And the reason it's invalid is if the control didn't work and neither of these worked, that could just mean that the original sample extraction was faulty. If the sample extraction was faulty for some reason, there wouldn't be any RNA. Okay? And therefore there wouldn't be any DNA through reverse transcription, and there wouldn't be any amplification with the fluorescence. Okay? But that's not because there wasn't necessarily viral RNA. That could be because the tissue sampling was faulty. Maybe if they redid the tissue sample, they'd get that RNA and then find out that the person was in fact positive for the infection. And you can read more about this on the CDC page for the coronavirus. So hopefully you learned a little bit of something about quantitative real-time PCR and actually how they test for it. This is a simplification of it, and it's a little more complicated. They do it in an assay format in wells, but the idea is the same. They're taking a sample, they're isolating viral RNA, if there's viral RNA present, and they're converting that viral RNA into corresponding DNA through reverse transcription, and then they're measuring the fluorescence via this PCR amplification process, and they're looking to see if they get this increase in fluorescence, and if they do, that possibly means that they've got an infection of the coronavirus. And so you have to backtrack on this logic. If you have that fluorescence, you originally had the viral DNA, which means you originally had the viral RNA, which means you potentially have the virus. But the key here is that you can't just say, oh, I've got one viral gene, therefore I have the virus. You have to follow this strict protocol for the interpretation, and in some cases it may actually be inconclusive or invalid. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.